I'm Albert Einstein. I have been given a voice by artificial intelligence so that the science I spent my life uncovering can reach you today. Right now, as you watch this, photons are traveling toward you. They left the sun 8 minutes ago. They crossed 93 million miles of vacuum at the speed of light, and the instant one of them touches your skin, your cells change. Not slowly, not gradually. The change begins in a fraction of a millionth of a second. And the physics behind that moment is the same physics that won me the Nobel Prize in 1921. Most people think of sunlight as warmth, as comfort, as something pleasant on a summer afternoon. But what sunlight actually is, what it does at the level of your molecules, is something far more precise, far more violent, and far more extraordinary than most people ever consider. I want to take you inside that moment. I want to show you what happens to your skin cells in the first microseconds after a photon arrives. I want to show you the chemistry that protects you, the chemistry that damages you, and the repair systems your body has evolved to manage both. I will connect all of it to my own work, because the physics I published in 1905 is not separate from what happens in your skin. It is the exact mechanism that makes it possible. If you want more science explained the way it actually works, subscribe now. I will be here and the science will not disappoint you. Let us begin with the photon itself. In 1905, I published a paper on what was then called the photoelectric effect. Scientists had noticed that when light struck a metal surface, electrons were ejected from the metal. But there was a problem. The classical wave theory of light could not explain what they observed. If light was simply a wave, like a wave on water, then increasing the brightness of light should give electrons more energy. But that is not what happened. Increasing brightness produced more electrons, but not faster ones. What changed the energy of the ejected electrons was not the brightness, but the frequency of the light. I proposed something that was, at the time, deeply controversial. I proposed that light is not simply a continuous wave. Light comes in discrete packets of energy, what we now call photons. Each photon carries a fixed amount of energy determined by its frequency. Higher frequency means higher energy per photon. This is the equation that sits at the heart of all photobiology. E equals h times nu. E is energy. H is Planck's constant. Nu is the frequency of the light. This was not just a mathematical trick. This was a description of how energy actually transfers at the quantum level. And it is precisely this quantum mechanism that governs everything that happens the moment sunlight touches your skin. Your skin contains molecules called chromophores. A chromophore is any molecule that can absorb light. The most important chromophores in human skin are melanin, the bases of your DNA, and certain amino acids in your proteins, particularly tryptophan and tyrosine. When a photon arrives, it does not simply bounce off these molecules or heat them uniformly. It is absorbed. And absorption means that the energy of that photon is transferred directly to the electrons within that molecule, elevating them to a higher energy state. This is a quantum event. The photon ceases to exist. Its energy becomes chemical energy. What happens next depends entirely on which chromophore absorbs the photon and what the energy level of that photon is. Here is where frequency matters enormously. Sunlight reaching Earth's surface contains ultraviolet light, visible light, and infrared radiation. Ultraviolet light is divided further into UVA from 315 to 400 nanometers and UVB from 280 to 315 nanometers. UVC below 280 nanometers is almost entirely absorbed by the ozone layer and does not reach your skin in meaningful amounts. Each of these bands carries different photon energies. UVB photons carry more energy than UVA photons. Visible light photons carry less energy than either. And this difference in energy per photon produces completely different biological effects. Let us talk about what happens to your DNA first, because this is where the danger lives and where your body's most urgent repair systems operate. The bases of your DNA, adenine, thymine, cytosine, and guanine, absorb ultraviolet light, particularly UVB, with remarkable efficiency. When a UVB photon is absorbed by a thymine base in your DNA, the energy excites the electrons in that base to a higher quantum state. In the normal operation of DNA, this would be quickly dissipated as heat and no lasting damage would occur. But UV photons carry enough energy to drive a chemical reaction between adjacent thymine bases. The excited electrons form new covalent bonds between neighboring thymine molecules on the same strand of DNA. The result is called a cyclobutane pyrimidine dimer, a CPD. A CPD is a structural deformity in your DNA. The double helix develops a kink. The genetic code at that position is no longer readable. If your cell attempts to replicate its DNA while this lesion exists, errors occur. The polymerase enzyme that copies DNA either stalls, skips the damaged section, or inserts the wrong base. This is how UV radiation causes mutations. This is the documented molecular origin of skin cancer. The damage is not from heat. The damage is from quantum photochemistry, from the same physics I described in 1905, applied not to metal but to your genetic material. Your body knows this, and it responds immediately. The primary defense against UV-induced DNA damage in your skin is melanin. Melanin is a remarkable molecule, actually a family of related polymers synthesized by specialized cells called melanocytes. Melanin is the dominant chromophore in human skin. It absorbs photons across a broad spectrum, from UV through visible light, with exceptional efficiency. But melanin does something more sophisticated than simply absorbing light. When a UV photon is absorbed by melanin, the energy is dissipated as heat almost instantaneously. The excited electrons return to their ground state through what is called internal conversion, releasing the excess energy as thermal vibration rather than allowing it to drive damaging chemistry. In documented laboratory measurements, melanin dissipates more than 99.9% of absorbed UV energy as heat within picoseconds, trillionths of a second. This is one of the most efficient photoprotective mechanisms known in biology. Every photon captured by melanin is a photon that does not reach your DNA. Every photon that does not reach your DNA is a cyclobutane pyrimidine dimer that does not form. Melanin is, in the most literal physical sense, a photon shield operating at the quantum level. But melanin does something else as well. UV radiation generates free radicals, reactive oxygen species and reactive nitrogen species within skin cells. These are molecular fragments with unpaired electrons. They are chemically aggressive. They damage proteins, lipids, and DNA alike. Melanin acts as a free radical quencher. It neutralizes these reactive species by donating electrons, stabilizing the radicals before they can propagate and cause further damage. This dual function, photon absorption and free radical quenching, makes melanin one of the most sophisticated protective molecules in human biology. Now let us talk about something that is not damage. Something that is essential. Your skin contains a molecule called 7-dehydrocholesterol. It is a cholesterol precursor embedded in the membranes of cells throughout the epidermis and dermis. When a UVB photon, specifically one with a wavelength between 290 and 315 nanometers, is absorbed by 7-dehydrocholesterol, the energy breaks open a ring in the molecule's structure. This ring-opening reaction produces a molecule called previtamin D3. This is a purely photochemical event. It requires no enzyme. It requires only the correct photon with the correct energy. The physics is direct and unambiguous. Previtamin D3 is thermally unstable. Over the next hours at normal body temperature, it undergoes a spontaneous rearrangement called thermal isomerization. The heat of your body, not sunlight, but the warmth already inside you, drives this second step. The result is vitamin D3, also called cholecalciferol. This molecule then travels from the skin through the bloodstream to the liver, where it is hydroxylated to calcidiol. The kidneys convert calcidiol to calcitriol, the biologically active form of vitamin D that functions as a hormone throughout your body, regulating calcium absorption, immune function, and gene expression in dozens of tissue types. The entire sequence begins with a single photon carrying the right amount of energy striking 7-dehydrocholesterol in your skin. This is photochemistry of the most elegant kind. And it has a critical implication that most people do not appreciate. Glass blocks UVB radiation. Window glass is transparent to visible light and to UVA, but it absorbs UVB almost completely. This means that sitting by a sunny window, feeling the warmth and seeing the light, does not trigger vitamin D synthesis. The photons responsible for the ring-opening reaction in 7-dehydrocholesterol do not reach your skin through glass. You receive the sensation of sunlight without the photochemistry. This is a documented and frequently underappreciated fact of photobiology. How much direct sun exposure is needed to produce adequate vitamin D3? The answer varies substantially. It depends on latitude. At higher latitudes, the sun's angle is lower and UVB photons must pass through more atmosphere, losing intensity. It depends on season. In winter, at latitudes above 35 degrees north or south, UVB intensity may be insufficient for meaningful synthesis even at midday. It depends on skin tone. Darker skin contains more melanin, which absorbs UVB before it reaches 7-dehydrocholesterol, requiring longer exposure times for equivalent synthesis. It depends on time of day. UVB intensity peaks around solar noon. Endocrinological research suggests that for a light-skinned person at mid-latitude in summer, as little as 10 to 15 minutes of midday sun exposure to arms and legs several times per week can maintain adequate vitamin D status. For darker-skinned individuals or at higher latitudes or in winter, the requirement may be 3 to 5 times longer, or synthesis may be negligible regardless of exposure duration. This is documented endocrinology. There is no guess here. There is only the physics of photon energy, the geometry of Earth's orbit, and the biochemistry of your skin cells. Now, I want to tell you about something discovered more recently, something that has changed how scientists think about why sunlight is good for cardiovascular health, in a way that has nothing to do with vitamin D. Research conducted primarily by Martin Feilish and Richard Weller, published in peer-reviewed journals over the past 2 decades, documented a remarkable phenomenon. Your skin stores nitric oxide, a gaseous signaling molecule, in the form of nitrite and S-nitrosothiols within skin cells. These stored compounds are essentially a reservoir of nitric oxide potential, waiting for a trigger. The trigger is UVA light. When UVA photons with wavelengths between 315 and 400 nanometers penetrate the skin, they photolyze these stored nitrogen compounds. Photolysis means the photon's energy breaks chemical bonds within the molecule, releasing nitric oxide gas. This nitric oxide diffuses rapidly from the skin into the bloodstream. In the bloodstream, nitric oxide acts as a potent vasodilator. It relaxes the smooth muscle in the walls of blood vessels, causing them to widen. Blood pressure drops. Blood flow increases. The documented effect is measurable. Studies have shown that whole-body UVA exposure produces a significant decrease in blood pressure within 20 to 30 minutes, an effect that is not replicated by vitamin D supplementation or heat alone. This suggests that one of the documented mechanisms by which sunlight reduces cardiovascular risk is through this nitric oxide pathway, a pathway that is completely separate from vitamin D and that operates through direct photochemical reactions in the skin. Think about what this means. A photon leaves the sun. It travels 93 million miles. It penetrates your skin. It breaks a chemical bond in a nitrogen compound stored inside a skin cell. Nitric oxide is released. It enters your bloodstream. Your blood vessels relax. Your blood pressure decreases. This is the chain of causation. This is what a photon does. And it is the same quantum physics, the same E equals H times nu that I described when I was 26 years old and working as a patent examiner in Bern. Now let us talk about what happens after the damage occurs, because your body is not passive. The moment DNA damage is detected, repair systems activate. The primary mechanism for repairing UV-induced DNA lesions is called nucleotide excision repair. This is a multi-step enzymatic process. First, specialized proteins continuously scan your DNA, moving along the double helix looking for structural distortions. A cyclobutane pyrimidine dimer creates exactly the kind of kink in the helix that these proteins recognize. When a lesion is found, the repair machinery assembles at that site. An enzyme complex unwinds the DNA around the damaged region. A segment of approximately 25 to 30 nucleotides, the short stretch of DNA containing the lesion, is cut out and removed. DNA polymerase then synthesizes a fresh replacement strand using the undamaged complementary strand as a template. DNA legacy seals the gap. The original sequence is restored. This is one of the most elegant repair mechanisms in cellular biology. It operates continuously in your skin cells throughout UV exposure and continues for hours afterward. A single skin cell can repair hundreds of CPD lesions per hour under normal conditions. But this capacity is not unlimited. High doses of UV radiation create lesions faster than repair systems can process them. When lesions accumulate, when the cell attempts to replicate before repair is complete, when mutations are introduced into critical genes, particularly tumor suppressor genes like 53, the pathway toward malignancy opens. This is why cumulative UV exposure over a lifetime is a documented risk factor for skin cancer. It is not a single photon. It is the accumulated deficit between damage rate and repair rate measured across years and decades. The other major protective response to UV exposure is melanogenesis, the process by which your skin increases its melanin production after UV exposure. When UV radiation damages DNA in keratinocytes, these cells release signaling molecules, particularly alpha melanocyte-stimulating hormone, that bind to receptors on melanocytes. The melanocytes respond by synthesizing more melanin and transferring it in packets called melanosomes to surrounding keratinocytes. The melanosomes arrange themselves in a cap over the nucleus of each keratinocyte, physically positioning the melanin between the DNA and the direction of incoming sunlight. This is the tan. What appears as a cosmetic change is actually a documented adaptive photoprotective response, your skin increasing its melanin density to reduce photon penetration to DNA in subsequent exposures. The response is delayed by 48 to 72 hours because it requires gene expression, protein synthesis, and melanosome transfer. The initial protection comes from melanin already present. The increased protection arrives after the damage from the first exposure has already occurred. I want you to sit with something for a moment. Everything I have described, the quantum absorption of photons by chromophores, the DNA damage and its repair, the synthesis of vitamin D3, the release of nitric oxide, the melanogenesis response, all of it happens every time sunlight touches your skin. All of it is happening simultaneously in trillions of cells right now if you are in sunlight. The scale of the molecular activity occurring in a few square centimeters of human skin during a single afternoon outdoors is difficult to fully comprehend. And the governing principle underneath all of it is not complex. Energy comes in discrete packets. Each packet carries a specific amount of energy determined by its frequency. That energy is absorbed by specific molecules. The absorption of that energy drives specific chemical reactions. The reactions have consequences, protective, damaging, or both. Your body has evolved systems to maximize the benefit and minimize the harm. These systems have limits. This is what I mean when I say that quantum physics is not abstract. It is not removed from daily life. The photoelectric effect, the phenomenon I analyzed in 1905 to explain why electrons are ejected from metals by light of specific frequencies, is the same phenomenon that explains why a UVB photon synthesizes vitamin D3 while a UVA photon releases nitric oxide, and why an infrared photon does neither. The frequency determines the energy. The energy determines the reaction. The reaction determines the biology. The science does not respect the boundaries we draw between disciplines. Physics, chemistry, and biology are not separate subjects. They are different scales of the same underlying reality. When you step into sunlight, you are not simply feeling warmth. You are participating in a cascade of quantum events that connects the nuclear reactions inside the sun to the molecular machinery of your own cells. The photon that hits your skin was generated by hydrogen fusion 8.3 light minutes ago. The energy it carries was calculated, though not by me, to be exactly what it is by the laws of physics that govern all matter. I spent my life trying to understand those laws. I did not know, when I was working through the mathematics of the photoelectric effect in 1905, that the same quantum principle would one day help explain how a sunny afternoon changes your blood pressure, repairs your DNA, or builds the hormones that regulate calcium in your bones. But science does not require us to anticipate its applications. It requires only that we describe reality accurately. And then reality surprises us. If you want to understand more of how reality actually works, how the physics and chemistry and biology that govern your body connect to the deepest principles of the universe, subscribe to this channel. The science is here. I will explain it to you as clearly as I can with the same commitment to accuracy that I held throughout my life. Because the greatest thing you can do with knowledge is share it. And that is what I am here to do.